Our next speaker is Anne Cochrane. She's going to be talking about plant responses to changing climates in the South Coast, particularly banksia species, I think. She's a senior research scientist with the Science Division of the Department of Parks and Wildlife. She manages the Threatened Flora Seed Centre, a long-term storage facility for seed of rare and threatened species of Western Australia. Her role is to collect, test, store and monitor this seed and ensure its viability for the future. Anne's work also involves research into new techniques for the collection and storage of seed. She's interested in plant-animal interactions, where this relates to seed set, seed predation and the productivity of species. She's currently undertaking research into the effects of temperature and moisture availability on seed germination and early seedling growth in banksia species. Over to you.
Thank you. Okay. This is, a lot of what I'm going to talk about is from my PhD that I completed at the end of last year. I was a late starter, and specifically plant regeneration. Now forwards. Oh yes, plant regeneration. Well, germination is really critical in the life cycle of plants, particularly those that are obligate seeders that are killed by fire and require seed for recruitment and persistence in the landscape. And germination is driven by temperature and moisture availability to the most part. Timing of germination is generally matched to the appropriate time for seedling growth and survival. So obviously when the climate changes, there are going to be big impacts on germination and therefore on plant diversity. And then as germination shifts, you'll get population dynamics changing, diversity of communities, abundance, all that sort of stuff, and distributions are likely to change as well. I'm going to do a quick romp through what's, sort of 4 years. So all this has been written up in papers. So if I lose you along the way, don't worry, you can read all about it. So I used a bunch of different approaches, field, laboratory and glasshouse approaches over the couple of years. I was interested in whether populations of species varied in their responses to the stress, that the drought and heat stress, and also did this show local adaptation. As you can see from, which is the, no, whoa, hey, that's not a good idea. There, oh, yes. Southwest Western Australia. It just so happened that I picked 4 species that occur along the south coast, and obviously it's warmer over here, we've heard about, and drier, and cooler and wetter here. The populations of the 4 species that I picked spanned from about Northcliffe here over to Cape Arid. They didn't necessarily overlap, but we have this nice correlation between longitude and mean annual precipitation and mean annual temperature. I have banksia coccinea, banksia baxteri, banksia media, and banksia quercifolia that I looked at in 6 populations of each of those species. I used a common garden experiment, thanks to the Great Southern Institute of Technology out at their Mercer Road site. I used rain-out shelters and open-top chambers that allowed me to drought stress and heat stress seedlings from these 24 populations. Rather a lot of seed was sown. The response to warmer soils was that warming was delayed and reduced seedling emergence. That was quite evident across 3 of the 4 species anyway. The species varied in their response, and there was variability in how the populations of each species responded. And this is just percentage emergence, and you see that this is control on the left-hand side and warm on the right-hand side. And so for 3 of the species, you saw this decline in emergence. You also saw, this is the mean time to emergence, that these are the in days, and as you warmed the soil, it took them longer to emerge. And this was just showing the difference in 6 populations of banksia quercifolia, and that they didn't all respond in the same manner. Laboratory experiment. This is a temperature gradient plate, courtesy of South Coast NRM. This is, again, 4 species, 6 populations, and I tested them across foot in constant temperatures. So there's 196 little cells here that gives you a nice gradient, so you can go from cold here to hot here, and it swaps over. I used it in a bidirectional mode, so you can get a whole range of different scenarios of temperature. And this is actually just a screening process because I can't replicate it, but it just gives you some idea of what's going on. And the response here, what we have here is this is the temperature window for 90% germination. We've got banksia, coccinea, media, and quercifolia here, and we can see, let's take banksia media, has rather a wide window for germinating up to 90%, and then you get banksia coccinea, which is much smaller. And then what you do get is this is the optimal germination, and either side it takes longer for these seeds to germinate. So it's possible for high germination, but you do get longer time periods to germinate. And here we have just showing the population differences. They don't all react in the same manner. This is percentage germination, and the mean time to germination also changes considerably. So we have this population variation. Another laboratory experiment where I used polyethylene glycol to drought stress seedlings at a couple of different temperatures. And this was another large experiment with over 10,000 seeds. And the response to drought stress was that germination again declined as moisture stress rose, and that was quite marked. And the time to germination doubled with the increasing moisture stress. And there was an interaction between temperature and moisture that it did influence germination, and again we see population variation. And these are just some graphs that just show you the differences at the different, here we've got the different temperatures, 10, 15 and 20, and how temperature actually does affect, this is the moisture stress, so this is just no moisture stress, and then increasing moisture stress, and how you actually get this quite sudden decline, and you get quite a marked increase in the mean time to germination. And again, here we see banksia media actually at its preferred temperature of 20 degrees. It's actually quite resilient to moisture stress, but when you put it at a suboptimal temperature, which is 10 degrees there, it declines markedly. But again, you see quite strong, just small amount of moisture stress, and it takes almost twice as long to germinate. I did a Glasshouse experiment. This is all done through the Australian National University, so this is over in Canberra now. And I used 7-month-old seedlings of both banksia and coccinea. I picked these two species because they do co-occur across their range. I used the 3 populations from the 3 different rainfall zones, and I had 2 temperature and 2 moisture conditions. So the response to warm, dry conditions here, what we see is significant declines in the seedling growth and fitness-related traits under the warm, dry conditions relative to those seedlings grown under cool, wet conditions. So just these quick graphs here, okay, banksia is in blue, coccinea is in red. These are the leaf traits. So what I've got here is leaf number, leaf mass, leaf area, leaf length, specific leaf area. This is a height growth increment and above ground biomass and chlorophyll content. And the 3 graphs represent, here's the change just with drought, here's just with warming, and here's with warming and drought together. So what you see here with drought is this is the percentage decline in these particular leaf traits. So the percentage decline here with drought is across all of the leaf traits except chlorophyll and the leaf mass ratio above ground. With warming, you don't get such a warming alone, you don't get such a decline. Some of these are like leaf number increased, the height increased. So you see, something quite different if you just put, these 2 separately. But if you put warming and drought together, once again you see more decline, and more so in the different species. This represents whether it's significantly different from each other or not. So there's obviously something happening with warming and drying conditions in terms of seedling growth. So I've got some summaries on this. So we have a declines in recruitment likely with warming and drying. So it's going to be a decline, and it's going to be slower germination. Now, if you have slower germination, especially in a winter wet, summer dry situation where we expect to have germination happening in autumn, winter, then if these species are going to be slower to germinate, then they're going to be closer to the summer dry, and therefore their roots aren't necessarily going to be down into the soil sufficiently for them to survive over particularly their first summer anyway. The species responded differently, so obviously you can't necessarily say, okay, well, a banksia does this, so all banksias are going to do this. I had 4 banksias, and they did all respond generally in different manners. Within species, their responses also varied, and this means that you can't just say when you're modelling that, species X does this, because species generally occur across a geographic gradient, an environmental gradient, and therefore in numerous populations that are subjected to different environmental stresses, and they react in different manners. I mean, they might not always do, but I've found that they do generally. So it can be misleading if you just say species X does, ABC. It also, from my point of view, highlights the need to conserve multiple populations across their range. So once again, for seed conservation, people feel that, if you've collected seed from a species, tick in the box, that's good enough. But no, we really need to conserve species conserve species in situ and ex situ across their range. And flexible species are likely to respond to change better. Now, I'll just, there was a lack of clinal response. What I didn't see is that the populations in the dry eastern end of the range were more tolerant of drying, warming climate. So it just means that we can't necessarily say, okay, well, pick the seed from the dry end of the gradient and use that in a warmer world, because it just doesn't work like that. And so really seed source selection is not going to be simple for restoration. What you really do need is to have a nice, local might be good in some respects, but probably, you want to collect across a species range to give species their best chance of survival in future climates. Now, this is just quickly on to some work that was done, in fact, before I started the PhD, and this is when South Coast NRM purchased this temperature gradient plate for me. I did screening of all the 38 obligate seeding banksia species from the southwest against current and future climates. This is all that. So that's how it works. It's cold here, it's hot here, it fluctuates here, and it's constant across here. This is the sort of graph that's produced where you have the day temperatures. This is just a parody of the actual temperature gradient plate. So you have day temperature, night temperature, and this is what the kind of graph I would get for banksia dryandroides, where we've got here yellow is 100%, down to the dark blue is 0%. You've got very little germination happening down here. You've got banksia media here, and germination is really high across all these different temperatures. So, I mean, this is where we might say, after screening, this is at risk, and this might be pre-adapted to a warming climate. This has just recently been accepted for publication. So the optimal germination timing, when I modelled it against current climates, 1950 to 2000, and then the 2070 high emission scenario of the RCP 8.5, what we saw is that the timing will shift in the future, about a month for most species. So maximum germination is likely, instead of occurring, say, in June, will occur in July, so that's pushing it more towards a drier summer. There didn't seem to be any differences really between threatened species and common species in terms of, you couldn't say, oh, well, threatened species are actually going to be at higher risk than some of the common ones. There's only one species, Banksia dryandroides, that had a major decline in germination. This is actually quite a useful tool to highlight conservation priorities. So although it's just a screening tool, what you'd do is you'd say, okay, well, there's Banksia dryandroides not doing so well. Now I'll start doing some really good, thorough work. And then it's empirical data for modelling. And I think that's all. I've got, the acknowledgements, obviously, of the South Coast NRM for the temperature gradient plate, which continues to be used up in Perth, and the Great Southern Institute of Technology, which provided this site for, which was quite a large experiment, and my colleagues. Thank you.